Hello, everyone. Good evening to all of you. I hope so. I'm audible to everyone, and the screen is also visible to everyone. Right? Am I clear? Just a second. Just a second. Yeah. Okay. Fine. Uh, so, uh, so today I welcome you all to the Brahmastra series, and we will be taking up the questions from Unit 6, that is Plant Physiology, and we'll be taking up the questions from right from nitrogen metabolism to stress physiology. Okay. Okay. Thank you for confirming, Gunashri and Sri Lakshmi. So, to begin with, the first question to all of you is regarding the nitrogen cycle. So, here microorganisms are given to you all, that is Rhizobium, Nitrosomonas, Nitrobacter, Pseudomonas, Azotobacter. Identify the correct pair, that means they are related to which phase of the nitrogen cycle. Good evening, Anusha. So we have different phases in nitrogen cycle. So all these are denitrification part, right? Then you are having the nitrogen fixation. Then you are having the nitrification part. What should be the correct answer for this question? Try on. This question I have taken from an old question paper, because there are chances that they may ask some relevant questions, and when it comes to nitrogen metabolism, usually we can options are not visible. Only okay, the screen is not visible. Okay, just a second, okay, I hope so now it's visible. Anusha. Okay, fine. Vijay Lakshmi, what about others? What about others? These are the microorganisms, and now you need to identify different phases of nitrogen cycle relevant to the microorganism. Find out. Okay, Gunashri and Vijay Lakshmi. Anyone else would like to give your input? Anyone else would like to give the input? Okay, let's get into the answer. The correct answer is, as mentioned by Vijay Lakshmi and Gunashri, yes, option 2 is the correct answer. Now, how option 2 is the correct answer? You need to identify the process. So, nitrogen cycle, we are getting nitrogen from the atmosphere, and this nitrogen can be fixed in different format by lightning, by physical, chemical, and biological process. So now here we are taking the biological process of nitrogen fixation. So what is happening here? We have specific nitrogen fixing bacteria that will be converting the nitrogen into ammonia, and this ammonia will be converted by certain group of nitrifying microorganisms into the assimilatory form like nitrite and nitrate. So, after the utilization by the plants, we have a group of denitrification bacteria that will recycle the remaining nitrogen back to the atmosphere. So, this is the overall process of nitrogen cycle, and ammonification, where there will be directly conversion of ammonium ions into nitrite. So, here also you are having some species like bacillus to carry out the process. So remember, the first step of converting atmospheric nitrogen to ammonia is carried out by diazotrophs. Nitrification can be classified into two major steps. First, it will be converted to nitrite by Nitrosomonas and coccus. Then it will be converted to nitrate by nitrobacter and nitrocyst. So now it is in the assimilatory form. And once they are being assimilated, remnants will be recycled back to the atmosphere with the help of Pseudomonas. So either you can remember in this format or you can remember in this format a flowchart image, whichever is convenient to you. But yes, it is important. And here we have a table also that is being given in your Teis and Ziegler book, where in the nitrogen fixation was known to be carried out by two types. One could be a symbiotic nitrogen fixation, another will be a free leaving nitrogen fixation. So the point to be noted over here is you have certain microorganisms which can carry out dual purpose. That means if you see nostoc, they can carry out fixation via symbiosis as well as free leaving. Similarly, just check it out, Anabina. Then you have azospiralium. So these are the microorganisms which can fix the nitrogen either with the help of the host plant or individually. Then another point to be noted over here is C. We have acetobacter, that is a symbiont for sugarcane. On the other hand, here we have azotobacter. So very minor difference between the terminologies. But this is an example of aerobic free-leaving nitrogen fixer. So be clear with the terminologies and also remember the microorganisms which carry out dual. Next question is, following are certain statements regarding the nitrogen uptake and assimilation by plants. Plant roots can take up nitrogen in the form of nitrate, and ammonia. Ammonium ions taken up by the plants can be directly assimilated into amino acid. These amino acids are synthesized exclusively in plastids and chloroplast of root and leaves respectively. Nitrate can be stored in the vacuoles of both root and leaves. Identify the correct combination of statement. This was a recent question. So basically, when I say nitrogen metabolism, they ask questions on rotational basis. That means sometimes they ask and sometimes they won't ask. Okay. So even if they will ask, it will be either in the form of this kind of statement based question, or it could be in the form of your match the following if they are speaking of organisms, microorganisms. Okay. Yes. Nature of Therapist and Gunashri has given the answer. Anusha also gave the answer. Have you read the statements properly? Yes. So the correct answer over here is, but as you mentioned, it's correct. It is your A, C, and D. So by mistakenly, it's given as 3. It should be 4. Because we have already seen that ammonium ions has to be converted into... No. See, this is correct only. See, the state option 3 is correct. Why? Because they can be taken directly by the plants and assimilated into amino acids. Now, that is a true statement. Now, whereas if you take into consideration the statement C, amino acids are synthesized exclusively in plastids and chloroplasts. Not necessarily, it can also be synthesized in the cytoplasm. Right? So we cannot say that exclusively in plastids and chloroplast. So we have cytosol as a location as well where the amino acids can be synthesized. That's why, so this is correct. Answer 3, A, B, and D. I hope so it is clear without any confusion. This is clear and ABD is correct. Right? 4 is incorrect. As I told you, the reason these are not exclusively produced in plastids and chloroplast. Okay. Okay. Next question is regarding the plant hormones. Now, this is a match the following kind of question. So, you have 3 columns. This is hormones, this is your receptors, and this is the response. Identify the correct combination here. Correct combination. So, through this question, they are trying to analyze whether you know about the signaling pathways of plant hormones or not. Because somewhere indirectly they are provided you with the response. So, they want to know whether the signaling part is clear or not. So, instead of asking directly, they have asked in a combination form. Yes, try out auxins and gibberellins. This combination. Transmembrane receptor. Response mediated by phosphorylation or dephosphorylation. On the other hand, cytokinin and racinosteroid. This is a soluble receptor, and the response can be mediated by proteasome mediated protein degradation. So you have to combine the three components together and remember what is happening or how the signaling is carried out. Is it tough? Do you find it tough? Okay, Malini, yes. I got your answer. Nature of therapist, okay. Gunashri, Vijayalakshmi. Okay, Anusha, 2 or 3. Why there is a confusion between option 2 and option 3? Try to recollect the signaling pathway. I guess you have created the signaling pathway in the form of flowcharts, or maybe you might be following the images. See, signaling and all, try to be in touch in a regular interval of time. The reason why I am saying is that there is a tendency of forgetting it. So when you keep on revising it on a regular basis, you'll come to know that yes, you are getting confident on that. At least, I'm not saying throughout, at least till the examination. Specifically, now you are on the verge of examination. Okay, Gunashri, Vijayalakshmi, Anusha. Anyone else would like to give the answer? Anyone else? Okay, you are going with 2 option. Okay. Anyone else? Fine. So the correct answer over here is 2. Now coming to this point, auxin and gibberellin, right? These are soluble receptors which can generate the response by means of proteasome mediated protein degradation. Can anyone tell me, when it comes to auxin, what is the degradation takes place, and when it comes to gibberellin, what is the degradation takes place? Here they are saying that response is mediated by proteasome mediated protein degradation. So some protein is getting degraded in that signaling pathway. Which protein is getting degraded in auxin and gibberellin signal transduction pathway? Absolutely correct, Malini. For gibberellic acid, as you said, it is your DELA protein. This serves as a repressor. Yes. Next, auxin. Who is the repressor of the auxin? So somewhere here they are trying to ask you the repressor, because in every signal transduction pathway you need to remember the receptor location. Then along with that you need to remember the transcription factor and then the repressor. The 3 important things when it comes to plant signaling. No. ARF is a transcription factor, Malini. I think you got confused. ARF is a transcription factor. IAA is your auxin. So how auxin? Be clear. You are given the partial answer. Actually, the answer should be OXIAA, that is your early response genes. Are you able to recollect? Because it can have two functions. It can serve both as an activator as well as a repressor, depending upon the situation. So when you don't have an auxin, this OXIAA will go and bind to your auxin receptor. That is, your ARF will be inactivated. But when the auxin comes and binds to the receptor, this will be degraded, so that your ARF will undergo dimerization and the transcription takes place. So that is about your auxin and gibberellin. Coming to cytokinin and brasinosteroid, so they are having transmembrane receptor, and the response is mediated by phosphorylation dephosphorylation event. Anything you remember regarding that? What is phosphorylation dephosphorylation event occurs with respect to cytokinin and brasinosteroid? You remember in cytokinin we have type A ARR and type B ARR. So both of them will be phosphorylated. Then only you'll get the cytokinin response. So dephosphorylation will terminate the response. Similarly, the case of brasinosteroid, you remember BES1, BZR1, phosphorylation, dephosphorylation, they are the one which triggers the pathway. I hope so you are able to recollect. See, basically signaling and all, it is best to study with diagrams. That will be more fruitful to you. And if you highlight the important points over there, it will be more easy for you to remember. Okay. Moving on to the next question. This is also related to your plant hormones. Now they have provided you with some receptors here, right? Which we are knocked out independently in Arabidopsis plant. So the plants we are named as like this. You have to identify the correct statement out of this. So this is a question where they are trying to ask you about the clarity regarding the receptors and the hormones. Find out the correct answer over here. So TIR1 is defective in ABA signaling, COI1 in gibberellin, GIT1 in auxin, PYL in JA, COI1 in JA, PYL in ABA, TIR1 in gibberellin signaling and GIT1 in auxin signaling. Unless and until you are clear with the receptors, you won't be able to give the answer. And always see, nitrogen metabolism can be avoided at one stage. You can keep it at last. But plant hormones is something you can always expect questions. There is no chance of skipping. You can get it in any form, maybe in the experimental form, or maybe they will ask you directly. Yes. What is the correct answer over here? TIR1, GID1, COI1, PYL1. All these are names of the receptor. Could be, Malini. Try out. I'll be telling you the answer. So you try out what should be the correct answer. And I will also give you the examples and names of all the hormones with their corresponding receptor. Try out, everyone. This is a recent question. Try out. Anusha, nature of therapist. Then Vijay Lakshmi, Gunashri. Not able to get into a conclusion? No one is able to get into a conclusion? Okay, Anusha has tried up and came with a conclusion. What about others? Okay, let me give you the answers. I feel this is too much tough for you. With even though you gave the late answer, but still I appreciate your answer, Anusha. You are absolutely correct. So, how can you identify which receptor is what? Make a comparison table of that, or you can go for this image to make yourself understand. See, as you can see, jasmonate, auxin, gibberellic acid. These are soluble receptors. Where exactly it is present? It is present in the nucleus. Okay. So, and you told TIR1, Malini, that was correct only. Auxin is having TIR1. Gibberellic, G for G. GID1, jasmonate, CoA1. When it comes to cytokinin and ethylene, they are present in endoplasmic reticulum. Okay. When it comes to brassinosteroid, abscisic acid, well, abscisic acid receptors, depending upon the receptor, it can be present in the cytoplasm. It can also be present in the membrane. So this is your BRI1, that is the receptor name. And here you are having GTG1 bar 2, that is the receptor for abscisic acid. PYR, PYL, RCAR. These are the receptors. That means the cytosolic receptor present for abscisic acid. When it comes to cytokinin, you have CRE1, ETR1. These are the receptors. So if you remember this receptor, even this image, it will be easy for you to remember where is the location and what is the receptor. I hope so now it is clear to all of you, because I got only one answer, that is from Anusha, and Malini has tried up, but others I did not get a response. So I hope so now it is clear to all of you. Now you will not forget what is the receptor, what is the ligand, where is the location and all. Right? Moving on to the next question. Following are certain statements regarding seed maturation. Seed maturation involves mainly the accumulation of storage products like starch, lipids, and proteins. A large number of chaperones, including the family of Lea proteins, plays a crucial role in the desiccation process of seeds. The moisture content gets reduced with the maturation of seed. ABA and gibberellin both are responsible for promoting seed dormancy. Here also you have to identify all the correct statements. See, there are questions that can be asked regarding the incorrect statements. So, what you can do, what you have to do, you have to read the question thoroughly and know what the question they are asking. Are they asking correct statements or incorrect statement? Okay, Nature of Therapist Gunashri Malini has given the answer. Others, and as I always say, try to justify your answer when you are practicing the MCQs. I'm not asking in the exam. If exam also you're doing, that's great. But while practicing, if you try to justify your answer, that will give you the clarity that whether you have your concepts are clear or not. Okay. Try practicing the questions in that way. Okay. Yes. Anyone else? Yes. Gunashri, Anusha. What about others? What about others? Anyone else would like to give the answer? Okay, Nature of Therapist and Malini, yes, you have given the reason also over here. So basically what happens is that we have abscisic acid and gibberellin serving as antagonistic to each other. Yeah, it is otherwise known as dormant hormone as well. Because if abscisic acid is promoting, yes, Anami is promoting dormancy, Then gibberellic acid is promoting seed germination. So both are opposite to each other. Got it? Yes, Vijay Lakshmi and Anami. So this, when you study plant hormones, remember there are some combination hormones which you have to be very clear. Okay? Because questions do ask in that format. Now, this is a figure given over here. The next question. The plant hormones auxin and cytokinin and their interactions plays an important role in regulating apical dominance. They have provided with an image over here, and we have three genes Q, Z, and M that represents the genes involved in phytohormone pathway. Looking into the image, you have to identify the correct statements from the above. X is an auxin maintaining the expression of Q and Z repressing the M. So here there is X, this is your Y, Q, Z and M. Three stages: intact plant, after decapitation, axillary bud outgrowth. Y is cytokinin promoting axillary bud growth induced by M. Decapitation activates X. Decapitation means the top portion is removed. X is a cytokinin that represses the M. Identify the correct answer from here. You all are able to see the image right properly. Okay, Vijay Lakshmi. I got an answer from Vijay Lakshmi. Okay, Malini. Anyone else who can interpret this image and explain what is happening over here? See, when it comes to plant hormone, plant physiology, be thorough with the images that is given in Tays and Ziegler, because unlike other topics, you take into consideration other units. From unit 6, whatever the questions come, it directly comes from the book itself. So if you're thorough with that, you can easily crack answers from here. And there is a good weightage when it comes to unit 6. Okay, Malini. Malini has provided some explanation and we need to look on whether you are correct. Then anyone else? Anusha. Anusha has also given the answer. Vijay Lakshmi gave the answer. Anyone else? Okay, you feel that X could be auxin. So that means that's the reason you considered statement A as correct. Okay. What about others? Anami, try it out. Vijay Lakshmi. Okay, Vijay Lakshmi already gave the answer. Anami, nature of therapist. Okay. So the correct answer over here is absolutely correct. It's 3. So cytokinins are responsible for modifying the apical dominance and promoting the lateral bud growth. How? See, the image is directly taken from the book itself. See, here you have got intact shoot apex. IAA is auxin. So as long as auxin is there, apical dominance or apical meristem will be there. There will be no axillary bud formation. So what happens? IPT gene will be off because IPT is responsible for synthesizing cytokinin. Whereas CKX gene will be on, that is cytokinin oxidase. What is its function? Responsible for the breakdown of cytokinin. But if you decapitate it, that means the top layer is gone. So obviously the auxin will not be there. So what happens? Cytokinin synthesis prevails, and the lateral bud growth will be happening over here. So at that stage, obviously you require more of cytokinin, so IPT gene will be on. You don't want your cytokinin to be broken down. So the oxidase enzyme will be off. Now, after reaching a particular stage, that means once the axillary bud has reached a particular stage, they will induce the formation of auxin again. So when auxin is produced again it repeats into the first level. That is, IPT gene will be off, cytokinin oxidase gene will be on. So this is how you need to relate it to the existing image. Okay. So try to study unit 6 with images to the maximum. This is just a reminder to all of you that we are starting with Josh batch from 1st of July. So once you are enrolled into this batch, you will be having 24 into 7 support system. You will be having access to all kind of study materials and concept notes for your revision. You will also have access to internships, some AIMNet, that is unit wise test series are also there. So you can also have access to over there also, and premium guidance via chat support, live sessions from the corresponding faculties. So if you want to know more about it, you can always call to our toll free number or you can visit to this link. Okay. Next, following are some statements regarding the plant hormone ethylene. Climateric fruits exhibits a characteristic respiratory rise before the ripening phase, whereas non-climateric fruits will be exhibiting respiratory rise before the ripening phase. In the leaf shedding phase, higher auxin level increases the sensitivity of abscission zone cells to ethylene. In the leaf maintenance phase, the higher auxin concentration increases the sensitivity of abscission zone cells to ethylene. Identify the correct combination of statements here. Now, when you read such question, one thing that will strike your mind will be that obviously out of this two, one has to be correct, and out of this two, one has to be correct, because the sentences are synonymous. When it comes to climatic, non-climatic, obviously you cannot say both the cases there will be increase in a respiratory phase. Similarly, the different phases of abscission is there. So in every phase, you will not find the same level. Okay, Malini, Gunashri, and Nature. And I really appreciate, Malini, you always try to give some explanation to the question. And that's a very good practice. Yes, go on, others. Absolutely correct. Yes, nature of therapist, that is a basiputal movement of auxin. You have two types of movement, acropetal and basipetal. Anyone else would like to give the answer? Okay, let me get into a conclusion. So the correct answer over here, as mentioned by Malini and Gunashri, option 2. Now, how we can justify this and why the statements B and C are incorrect, we will have a look onto that. Remember, climateric fruits will ripen fully if they are harvested at completion of their growth period. On the other hand, non-climatic fruits will be ripened fully only if they are allowed to remain attached with their parental plant. So if you look into a comparison over here, see, the respiration rate for climateric fruits will be higher compared to non-climateric fruits. Ethylene production similarly for climateric fruits will be higher. Continuous ripening after harvesting, but here minimal ripening after harvesting. That's why it is mandatory that you should keep the fruit in the plant itself. Rapid changes in the color, texture, and flavor. Here the changes will happen, but only if they are present in the plant. Can be harvested before fully ripened. As I told you, after plucking out also it will be ripened. But this one typically harvested when it is fully ripened. So examples includes apple, avocado, banana, fig, mango, olive, peach. All these are climatic fruits. Non-climatic fruits include bell pepper, cherry, citrus, pineapple, strawberry. They can be ripened, but it will take a long process. So it's better to stay in the plant itself, and once they have ripened, then you can pluck it off. Okay, so that is the explanation. And next is the abscission zone, how it is being regulated. As you can see, we have three phases: leaf maintenance phase, abscission induction phase, and the abscission phase. In the leaf maintenance phase, you will find higher level of auxin is present. So at that time, ethylene sensitivity will be not there. That's why you can find green in color. When induction of abscission takes place, there will be gradual reduction in the auxin and gradual activation of ethylene. When the stage comes, we call it as an abscission phase. There will be synthesis of enzymes that will help in the removal of the leaves, and the leaves has completely yellow in color. So at this stage, ethylene level will be at peak and there won't be the presence of auxin here. So that's why the statements B and C were incorrect. I hope so you got the answer. You understood the answer, why that answers were incorrect. Right? Moving on to the next topic, that is sensory photobiology. Following are the statements regarding phytochrome interacting factors, a family of proteins that regulate photomorphogenic responses in plants. PIF promotes photomorphogenesis by serving as transcriptional activators of dark induced genes. This PIF, on interaction with PR, gets phosphorylated, followed by degradation via proteasome complex. Degradation of PIF takes place in the presence of light. PIF induced genes are not expressed in light. Here also, they are asking you the combination of correct statements. Now, this is a very important topic, sensory photobiology. When I say it's a very important topic from examination point of view, and the fact is that this topic can easily make you confuse in case if your concept is not clear, if your base is not clear. And they are literally asking those kind of questions only, confusing questions only. If you go through previous year question papers, you hardly find a direct question. Like, even if you find direct questions, you will be literally confused in case if you have not studied properly. So this particular topic requires conceptual clarity. Identify the correct combination of statements here. First thing is that you have to be clear. Phytochromes exist in two forms: PFR form, PR form. You need to correlate it with the transcription factor, that is PIF. And this is a transcription factor of which plant hormone? Can anyone tell me? PIF is a transcription factor related to which plant hormone? You told me ARF is related to auxin, right? PIF is related to which plant hormone? Yes, Malini, I got your answer. What about others? Okay, Anami. Meanwhile, can anyone tell me which plant hormone will have PIF as their transcription factor? Only two answers. What happened, Vijay Lakshmi? Gunashri, nature of therapist. Okay, Vijay Lakshmi and Anusha. So got four answers. Anyone else? Gunashri and nature of therapist, would you like to take up this question? Okay, great, Gunashri. So let's look on to the answer. What is the correct answer over here? The correct answer over here is 2. Now, why it is 2? 2 indicates A, C, and D. PIF promotes scotomorphogenesis by serving as transcriptional activator of dark induced genes. PIF on interaction with PR gets phosphorylated followed by degradation to proteasome complex. Now that is incorrect. Degradation of PIF can be taken place in the presence of light. So we can say PIF induced genes are not expressed in light. So C and D are somewhere correlated. If you're reading the sentences carefully, C and D are correlated. The only thing is that you have to identify the correct definition from A and B. So for that, look into the signaling pathway. So remember, we have two phases. One is the dark phase, another is the light phase. So dark phase, you have high gibberellic acid that leads to long hypocotyl generation. So phytochrome will be present in the PR form, that is the inactive form. So when your gibberellic acid enters inside and binds to the corresponding receptor, your phytochromes are not coming inside here. They are inactive and present outside. GRAS and DELA will be recruited, which is a repressor, and they will be targeted for ubiquitin mediated, proteasome mediated degradation. So what happens? Your transcription factor PIF is free, which can interact with the specific G box elements present within the DNA via BHLH domain, that is basic helix loop helix motif, and they carry out the gene expression. On the other hand, what happens? That is the light phase wherein your phytochromes are active. Because of which what happens? It can enter inside the nucleus and they will start targeting the transcription factor for degradation. Not only that, we know that our DELA and GRAS are also active during the daylight. So they will also target the transcription factors for degradation. So if transcription factors are gone, there won't be any gene expression and you will not get any kind of cellular response here. Okay. So this is one part. Another image you can see over here. Same thing, only representation is a different one. In the light phase, your phytochrome will be coming inside. Your PIF will be phosphorylated. So it will be ubiquitin proteasome mediated degradation. DELA will also combine with it. So there won't be any gene expression, see, preventing the expression of cell expansion genes. On the other hand, during the dark phase, what happens? You have got the gibberellic acid bound to your corresponding receptor. DELA will be recruited and it will be degraded. So depending upon the requirement, either they can go for cell expansion genes or they can block the chlorophyll biosynthesis gene. So that depends upon their requirement. Okay. This is how you need to understand and get into a conclusion to this kind of questions. Next, Following are some statements regarding jasmonic acid in plants. 12-oxo-phytodienoic acid is produced in chloroplast and transported to peroxisome. Action of lipooxygenase, allen oxide synthase, allen oxide cyclase will take place in peroxisome. 12-oxo-phytodienoic acid is first reduced and converted to jasmonic acid by beta oxidation. Final production of jasmonic acid takes place in chloroplast. Action of allen oxide synthase, allen oxide cyclase takes place in chloroplast. Here also you need to identify the correct combination of statements. Identify the correct combination of statements from here. So, jasmonic acid basically comes under secondary metabolites because you know that jasmonic acid and salicylic acid are released by the plants in terms of defense purposes, unlike the other five major plant hormones, that is your auxin, gibberellin, cytokinin, abscisic acid, ethylene, and brassinosteroids. Unlike those plant hormones, these are exclusively for defense purpose. So it's important to remember the jasmonic acid signaling pathway to get into a conclusion here. If you know the answer, you may say. If you don't know the answer, you can say that I can directly show you the answer and I will help you out in relating the question to the answer. Tell me whatever you want. And one more thing to be added over here, unless and until you have studied, if you have studied, then only attempt the question. If you have not studied, don't try to attempt because these are 4 marks question and you know very well. You will lose 1 mark if you are doing a guesswork. No problem, Malini. I will explain you out how it happens. Okay, nature of therapist. Okay, Anusha has given a trial for giving us 4 as the answer, 1, 3, and 5. Okay. Anyone else? Vijayalakshmi, Gunashri. Okay, that much you know that it is produced in peroxisome. Okay, no problem. I'll show you the image. The things will be more clear to you. So the correct answer over here is absolutely correct. Anami and Anusha, 1, 3, and 5. So action of these enzymes takes place in peroxisome is incorrect. Final production takes place in chloroplast is also incorrect. So what is the correct answer? Remember, jasmonic acid is the only plant hormone which is having lipid as a precursor, that is linolenic acid. And this is the pathway. You have 2 organelles involved in the jasmonic acid production. In the presence of lipooxygenase, allen oxide synthase, and cyclase, you are getting 12-oxo-phytodienoic acid. Where are these enzymes located? In the chloroplast. Now, this dienoic acid will move to the peroxisome, which will undergo beta oxidation 3 times to get jasmonic acid. So, the final steps happens in peroxisome. Now, if you look into the signaling pathway, remember, they require a combination of isoleucine to get itself activated. So, in the presence of JAR1, your jasmonic acid will be activated and recruiting SCF. What does this SCF will do? It will polyubiquitinate the repressor so that your transcription factor is free and this results in transcription. Okay. So now if you look into this image, you can see that jasmonic acid, isoleucine combined together binds to the COE1, that is your receptor. SCF will be a part of it, which will in turn polyubiquitinate the JAS protein. leading to degradation. So MYC2 becomes free, and this in turn leads to the transcription of jasmonic acid genes. I hope so. Now it is clear to all of you. Vijay Lakshmi, nature of therapist, Malini. And more clear for Anami and Anusha, although you have said it correctly, no doubt in that, but you are more clear with these images. Right? Okay. Moving on. I hope so you all understood it properly. Moving on to our next question. This is a kind of match the following. Identify the correct combination. So secondary metabolites again is a topic from where you can expect match the following as well as statement based questions. So this is an example of a match the following. Recently asked question. Phenolics, alkaloids, terpenoids, cyanogenic glycoside. Their structural unit. Isoprene unit, glucose unit attached via O beta D glucosyl linkage, nitrogen containing aromatic arene ring with OH group. Okay, Malini and Anami. So quick response. Any other answers? Any other answers? Remember to understand the structure of each of these secondary metabolites, because your secondary metabolites is broadly classified into these three components. Okay, and cyanogenic glycoside somewhere comes within the alkaloids itself, a part of it. Be clear with the structure, be clear with their function, be clear with their examples. That is very important. Okay, looking into the answer as given by all of you, it is 4, and yes, you are absolutely correct. I guess you all might be sure terpenoids is a 5 carbon isoprene unit. Phenolic compounds will be having aromatic ring. Alkaloids will be a nitrogen containing compound. So what is left? Cyanogenic glucoside. So you can easily click over here. Right? So this is how you need to approach the question. Phenylalanine, ammonia lyase, and chalcone synthase are involved in the biosynthesis of phenolic compounds in plants. Following are the statements regarding the action of PAL and CHS. Substrate for PAL and CHS are phenylalanine and chalcone respectively. PAL converts phenylalanine to tranexamic acid, or it converts phenylalanine to coumaric acid. P-coumaryl coenzyme A is converted to chalcone by chalcone synthase. Identify the true statement or the correct statement from here. Now this is PAL, CHS. It is related to phenolic compounds biosynthetic pathway, right? So in the session where I had mentioned that you have to be clear with their structure, function, and examples. Along with that, you have to be clear with their biosynthetic pathways as well. Which pathway belongs to which secondary metabolites? You might have heard about malonic pathway, shikimic acid pathway, mevalonate pathway, methyl erythritol pathway. All these things you have to be very clear. So as of now, I'll be showing you the phenolic compound related pathway. And let me see how many of you are able to give the answer here. Okay, nature of therapist has given the answer. Anyone else would like to answer here? So I'm getting a miscellaneous response. in the form of like a tug of war between option 2 and option 4. The answer will be correct if you are confident with the biosynthetic pathway. So let's check out the answer. Congratulations, whoever has given 4 as the answer, but not to be worried for those who has given the incorrect answer, because right now you will be corrected. See, phenylalanine in the presence of PAL will give rise to tranexamic acid. Okay, this gets converted to coumaric acid, coumaroyl coenzyme A, which in the presence of chalcone synthase produce chalcones. And this chalcones will be a precursor for flavanols, dihydroflavanols, anthocyanins, and tannins. Remember, from flavanols, you are getting flavones, isoflavones, and flavanols. Flavanols, you get it from here. Coumarins, you get it from coumaric acid. Benzoic acid derivatives, you get it from tranexamic acid. And lignin precursors, you get it from again coumaric acid. So this is important. If you remember this, you get an answer. Following are certain statements regarding plant pathogen interactions. Hemibiotrophic pathogens are characterized by initially keeping the host cells alive, followed by extensive tissue damage during the later part of infection. Effectors are molecules present in the host that act against pathogenic attack. Plants possess PRRs that perceive the MAMs in a specific class of microorganisms absent in the host. Phytoanaxin production is a common mechanism for resistance to pathogenic microbes in a wide range of plants. Identify the combination here. Okay, no problem, Anusha. I hope so. Now the things are more clear to you. Very good, Malini. Yes, you are correct. If you are giving a wrong answer here, somewhere you are getting corrected. So instead, never get disappointed with that. Be relaxed and think that, yes, I am rectified with this. And I will not make the same mistake again if I come across this kind of questions. Right? So that is the positive attitude that you must carry on with you. Not today or tomorrow, but always. Yes, what should be the answer here? Any idea? So we are left with very few time, and I would like to finish off the questions quickly. Okay, I got one answer, nature of therapist. So if I get one answer, I'm just displaying the answer because we are running time constraint. If anyone would like to take up the question, you are most welcome. Okay, Anami. So here the correct answer is option 2. So why option 2 is correct? See, option 2 is A, C and D. That means A is correct, C is correct, D is correct. Why B is incorrect? Any idea? The reason is that effector molecules are secreted by pathogens, not the host plants. Very simple question. And these molecules are designed to manipulate the host cellular process and facilitate infection or suppress the host defense mechanism. That is the point. So if you understand just one definition of effector molecules, you can easily have 4 marks with you. Once inside the plants, pathogens use one of the 3 main attack strategies. Necrotrophic pathogens means it causes complete tissue damage. Biotrophic pathogens means pathogenic attack with less damage. Hemibiotrophic means they cause severe damage, cause cell damage leading to even necrosis. That is the fact. Okay. Quickly moving on to the next question. Light is an important factor for plant growth and development. So we have several photoreceptors in higher plants like Arabidopsis thaliana involved in the perception of various wavelengths of light. So here are some statements. Red light photoreceptors represented by a gene family. Phytochrome C, the most prominent photoreceptor to receive the red light. Cry1 and Cry2 evolved from bacterial lyase. Far red light is perceived by phytochrome D. Identify the correct combination here. So you have to be clear with the different types of photoreceptors. Just two more questions to go and we will wind up the session. Anyone would like to take up the answer? Let me give you the answer. So the correct answer over here is A and C. So red light photoreceptors are represented by a gene family and far red is perceived by phytochrome D. So why this B and C are incorrect? A and C are correct. These two are correct. B and D are incorrect. So why so? Let's have a look. In plants, the most prominent photoreceptors for receiving red and far red light are both phytochrome A and B. Secondly, phytochrome C is not the prominent receptor. In fact, it requires other phytochromes for their stability and function. Option D is also false because far red light is perceived by phytochrome A. So we get into a conclusion of statement A and C. Now this is a kind of match the following: water deficiency, salinity, flooding, high light intensity, along with their effects. So this is related to stress physiology. Stress physiology and nitrogen metabolism are topics from where you get rotational basis kind of questions. That means you cannot expect always, but sometimes. Now this is very easy question even if you have not studied stress physiology. So the correct answer over here is 2. As you can see, high light intensity that is related to photosynthesis, it can cause photoinhibition. Water deficiency, it can cause hypoxia to the roots. Salinity, that is ions, sodium, ion cytotoxicity, and flooding can cause water potential reduction. Okay, so that will be the correct answer. You can go through the explanation later. And these are regarding the osmotic stress happening in plants. So quickly moving on, accumulation of ions during osmotic adjustment is predominantly restricted to vacuole. In order to maintain the water potential equilibrium within the cells, we have compatible solutes present. There are mainly 4 groups of molecules that serves as a compatible solute. So here the answer 3 is correct. I mean the statement 3 is incorrect. Why? Because galactose is not an example of an osmolyte. We have 4 major osmoprotectant: mannitol, sorbitol, glycine, betaine and proline. And this is the last question. This is regarding solute transport. Only reducing sugars are translocated, non-reducing sugars are translocated, sucrose and raffinose are translocated, D-glucose and D-fructose are translocated. Identify the correct statement. Be clear with reducing and non-reducing sugars with examples. Now you got a conclusion. So these two are correct, right? You can go through this explanation later, how the transport takes place. That's it. We have winded up with the session. And I wish you all the best for the upcoming exam. Study properly, do more of revision and practice more of MCQs. Now that is what important at this junction, because when you are approaching the exam. So once again, thanking you all so much and wishing you all the best.